Okay, so now we get this probable transporter table, and these are high confidence predictions where it thinks, yeah, I really have found a transporter. And what we need to do then is go through and review all of these. And in the same way, well, similar to what we did before with reviewing possible enzymes, we left click here. It tells you a bunch of things, and you can accept, reject, defer, leave it alone. I'm just going to go through and accept these. And so I'm going to go through and accept the rest of these, and then we'll come back and look at what we do next. Okay, so I went through, accepted all of them. I'm being very accepting today. You don't, of course, necessarily want to accept all of them. You'll actually be looking at the literature, relying on your expertise on this particular organism, etc. Now we've completed transport identification parsing. Refine. Other than that, we can update the overview again. Before we do that, let's just go through and be cautious and save the KB. It's good to save every so often. And now that we've saved, let's look at refine again. We can update the overview. Let's do that. And of course it'll bring up this thing saying, are you sure you want to upgrade the overview? There can be delay of an hour or more. It's not telling the truth. The delay is usually of 5 to 10 minutes. We just like to be cautious. And I'm going to say yes. And we'll come back once the overview is done updating. Okay, so now we have a sort of draft version of our model organism database. We've taken our GenBank annotation, fed it into pathologic, predicted gene products, predicted reactions, predicted metabolic pathways, predicted transport reactions, or more accurately identified transport reactions, predicted transcription units, associated compounds for the reactions, etc. We have our draft database. Now we go on to the next step of the pathway hole filling. Now the stuff you've done so far should have gone pretty fast. It went pretty fast for us, as we've seen. The pathway hole filling step is the step that takes a little longer. It's more of the set it up and do other stuff while it's running kind of step. You know, it won't take forever, take a couple of hours, but it's not a sit and watch step. There'll be some manual things you're going to want to do at the end. But between starting it and the end, you're just going to walk away from the computer or run it in the background or do whatever. Okay, so once again, we're on our database here, Coxiella burnettii, and we're going to go up and click on Tools and start Pathologic again. Just maximize that. And we're going to click Refine, go down to Pathway Hole Filler. Now you see here we have 3 options. The 2 I'm going to focus on here are Fully Automatic and Wizard. Fully Automatic is if you're just willing to accept a bunch of cutoffs, you're just going to take what it says. You don't want to manually look at the pathway hole filling. You probably don't want to do that in most cases because you're probably doing just the one database for the thing you just sequenced and annotated. You've done months of work on it. You're going to want to take a little time. So then you're going to want to use the wizard setting, which will kind of walk you through this. There's also the power user setting, which can be helpful if you've had to stop in the middle of the process or just you find you're a more advanced user and you want to tinker with individual steps. But we're going to choose wizard right now. So now we get this neat dialog box that tells us what we're going to do. It's going to tell you that the wizard consists of 3 main phases. We're going to plan, then we're going to perform our pathway hole filling, then we're going to review the predictions and decide which predictions we want to keep and which we don't. And as you'll see when we get there, it actually gives us a list of predictions. For a given pathway hole, we might have no predictions, we might have 1 prediction, we might have 5 predictions that beat a minimum quality cutoff, and then we'll be given a score for each one so we can decide, well, this is the highest scoring one, I like it, or maybe in some cases this is the second highest scoring one, but for reasons of my personal expert knowledge about this organism, I believe this one instead. I'm going to take that. So now that we've read this, let's click OK and go on to planning the analysis. Okay, so it says required BLAST protein data is missing but can be generated now if you wish. This process takes on the order of an hour. So we're going to need to do this. We're preparing BLAST protein data. Click on that. Okay, so it says choose known C. burnettii Dugway 7E912 reactions to use for training data. And that's known reactions are those reactions that have an enzyme assigned. So basically it's going to look at our set of reactions where we already have an enzyme assigned to that reaction, and it's going to use that as its training data set when it later goes along and starts blasting stuff back against our genome to work out other predictions for the pathway hole filler. And again, if you want the full, detailed, less kludgy explanation of how that works, you want to look at our publications. They're listed on the website. So here you have the option of selecting all, deselecting all, or entering a number of items to select at random. Now let's just click deselect all, and that clears everything. Now I had it set to select 100 items at random, but let's just select all. Let's be thorough. Okay. And then I'm going to hit OK, and this is going to choose these known reactions. It's going to use them for making our BLAST training data set, and that is, as it says, going to take on the order of an hour. And it's on the order of because we let you choose how many. You might pick 400, you might pick, well, 10 would be a bad idea, but you can pick anywhere in the range. Okay, so I'm going to click OK and let the process get started. Then it's going to ask me to choose a set of pathways or reactions to make predictions for. Basically here you can go with all pathways with holes, which is what I recommend if you're making a new pathway genome database, or you can select the pathways from a list or select the reactions from a list. And that's if you are going back maybe and doing refining and you just have one or two pathways you want holes filled for, just one maybe, or a couple of reactions. Maybe you've updated some things, you have reactions, you'd like some predictions just for those reactions, you can do that. But for now, since we're making a new database, we're going to go with the pre-default selected option, all pathways with holes, and say OK. Then it tells you when you click OK, the wizard will identify and evaluate pathways to fill pathway holes, and this could go on for a while. And at the end of this, you'll be prompted to review the predictions. So I'm just going to click OK and then let it run. And we'll come back once it's finished running and take a look at what we're presented with. And you'll notice as it's running on your machine that you'll have a progress bar. It'll show you what it's doing. It'll tell you what's going on. You can hit cancel if you need to. We're not going to do that now. We're going to let it run, and we'll rejoin this process once it's run through, and we can start evaluating and deciding which one of the pathway hole filler predictions we want to keep. Okay, so now Pathologic is done making its predictions, and it's time for us to go through them. You'll notice at the top it says minimum probability cutoff, 0.9. You can actually set that probability cutoff higher or lower. Once you've played with it, you'll get a feel for sort of what level of probability you trust from Pathologic. And you can redo the display to show things after you set the new cutoff. In our case, we're going to stick with 0.9. It's a good number. And now we have an option right up here at the top to fill hole with top candidate for all of them. That means you just take Pathologic's best guess for every single candidate. Now that's useful if you're trying to do this in a high throughput fashion and you don't really feel like manually examining every single candidate. In this case, since we're thinking more along the lines of making a database for our specific organism we're researching, we're going to look at each candidate individually and decide whether or not we like them for that spot.